Okay, so welcome to this next video in which we are discussing the vascular endothelial growth factor receptor. Okay, so we've discussed that when you have a portion of cells that are hypoxic, they will start releasing vascular endothelial growth factor A molecules. And we've discussed that there are 8 different isoforms of vascular endothelial growth factor A. Okay? And vascular endothelial growth factor A molecules will diffuse out in the interstitial fluid, and it is going to reach endothelial cells that line capillaries that are nearby this angiogenic center. Okay, and on the surface of these endothelial cells at the capillaries, you have a receptor for vascular endothelial growth factors, which is the vascular endothelial growth factor receptor 2. Okay, and we've discussed the structure of this vascular endothelial growth factor receptor 2. You have 7 of these immunoglobulin-like domains, a membrane-spanning alpha helix, then a split tyrosine kinase domain with a kinase insert domain between the two portions of the tyrosine kinase, and then we have this C-terminal tail down here. Now, what's going to happen is the homodimer of vascular endothelial growth factor A molecules is going to come and bind to 2 vascular endothelial growth factor receptor 2s. Okay, so let's show this, and it will cause the dimerization of the vascular endothelial growth factor receptor 2s. So let's draw out our vascular endothelial growth factor receptor 2s, both of them that we're going to now need. So here are these 7 immunoglobulin domains, and basically what's going to happen is the vascular endothelial growth factor A molecules, okay, which remember are existing as this homodimer, are going to link the 2 vascular endothelial growth factor receptor 2s together. Okay, so they will bind to the extracellular domain, and what is in the extracellular domain? We have these immunoglobulin domains. Okay, and by binding to both of the vascular endothelial growth factor receptor 2s, what the vascular endothelial growth factor A homodimer does is it dimerizes the 2 receptors together. Okay, so here are the transmembrane regions, which are now sitting in the lipid bilayer here. And then on the cytoplasmic side, you then have the 2 halves of the tyrosine kinase, with this kinase insert domain between the 2. And here is the carboxylic acid terminus of the protein, and then we'll add in the amino termini of the vascular endothelial growth factor receptor 2 proteins. Okay? And now just to complete the cytoplasmic domain of this side as well. So what happens is they dimerize together. Now also what's going to happen when the vascular endothelial growth factor A homodimer binds to the receptors is it's going to trigger conformational changes in the receptor that are going to lead to the dimerization of the 2 halves of the tyrosine kinase domains. Okay, so what's going to happen is these 2 halves of the tyrosine kinase are going to dimerize together. So let me redraw this. So I won't draw the entire extracellular portion again. I'm just now going to concentrate on the cytoplasmic portion. Okay, so I'm going to cut off those 7 immunoglobulin domains, immunoglobulin-like domains, and just now draw the cytoplasmic side, because they're not going to play any further role. It's the cytoplasmic side that is now interesting. So the 2 halves of the split tyrosine kinase will come together to make a functional tyrosine kinase, like so, and this will happen in both cases, so I'll draw it for both of them. So again, here is one of the halves of the split tyrosine kinase, and here is the second domain of the split tyrosine kinase, and here is the carboxylic acid terminus. And these little loops that we now have here are the kinase inserts. Okay, so we now have 2 functional tyrosine kinase enzymes. One is here, and the other is here. And basically, both vascular endothelial growth factor receptor 2s have tyrosine residues on their cytoplasmic side. So let me show you the structure of a tyrosine residue. Okay, and then I'll show you how we can phosphorylate tyrosine residues, which is the action of tyrosine kinase enzymes. And I should just write this down. These purple things are tyrosine kinase enzymes now. Okay, and they're functional, and they're going to phosphorylate tyrosine residues. So let me show you the structure of a tyrosine residue. So here's the amino group, which will be bound to the carboxylic acid group of the amino acid in front of it. Here's the alpha carbon with a hydrogen coming off. Here's the carboxylic acid group, which will be bound to the amino group of the amino acid after it. And then the R group of tyrosine residue consists of a methylene group, like so. Then with a benzene ring off the methylene group, here, and then off the benzene ring you then have an alcohol group. So this is the structure of a tyrosine amino acid. Okay, and for short, tyrosine amino acid residues can be denoted as Y. Y is the single letter amino acid code for tyrosine. Okay, so what we're going to do is we're going to now phosphorylate the tyrosine residue. Okay, and this involves the addition of phosphate groups. Now I'm going to show you it as though we're adding a pure phosphate group on, but in reality you'll be transferring the phosphate group from an ATP molecule. Okay, but I'll show you it as though we're adding on a pure phosphate. So the structure of a pure phosphate group, or an inorganic phosphate group, is as follows. You have a phosphorus atom at the center, and this phosphorus atom is double bound to an oxygen atom. And the phosphorus atom then has two alcohol groups coming off it, and also a single bond to a final oxygen atom, which then acquires another electron by ionic means, and therefore ends up with a negative charge. Okay, so this is a phosphate group. So basically what you can do is you can bind phosphate groups to alcohol groups via what is known as a phosphoester link. And let me try and explain this to you. Imagine for a moment that this phosphorus atom at the center here is not a phosphorus atom, but is instead a carbon atom. So imagine replacing that phosphorus atom with a carbon atom. Now ignore the fact that it has 5 bonds, which makes no sense if that's a carbon atom. And just look at this structure here, where we've got a carbon atom double bound to an oxygen with an alcohol group coming off it. That's the structure of a carboxylic acid group. Now, carboxylic acid groups can interact with alcohol groups and can form ester links. And what happens in an ester link is that you take off the alcohol group of the carboxylic acid group, you take off the hydrogen off the alcohol group, okay, you combine those together to make water, and you then link the oxygen to the to the carbon atom, as we're pretending it is, and that is then called an ester link. Now the exact same thing can happen when this is a phosphorus atom, basically, only the link that you now create is not called an ester link, but instead is called a phosphoester link. Okay, so we're going to phosphorylate tyrosine residues by forming a phosphoester link between the alcohol group of the tyrosine residue and a phosphate group. Okay, and as I say, this reaction won't occur with a pure phosphate group like this. Instead, you'll transfer it off the ATP molecule to create ADP. Okay, but it's the same principle, basically, that you're creating a phosphoester link. Right, so that's the reaction that these tyrosine kinases are going to catalyze. Now, which tyrosine residues are they actually going to phosphorylate? Well, basically, each one of these cytoplasmic domains of the vascular endothelial growth factor receptor 2s has tyrosine residues within it. Now, there are tyrosine residues in the kinase insert here. There are also tyrosine residues within this second half of the tyrosine kinase domain. And finally, there are also tyrosine residues in this C-terminal tail here. And in each of them, in the kinase insert, this second half of the split tyrosine kinase, and also the C-terminal domain, there are 2 tyrosines that are going to be phosphorylated, and this will go for this one as well. So you'll have a tyrosine here, 2 tyrosines again in the second half of the split tyrosine kinase enzyme, and then 2 tyrosines also in the C-terminal tail over there. And basically what's going to happen is this tyrosine kinase here that I've circled in pink, let's call this receptor 1, and let's call this receptor 2. Basically, the tyrosine kinase of receptor 1 is going to phosphorylate the tyrosines on receptor 2. So it's going to phosphorylate these two tyrosines in the kinase insert. It's going to phosphorylate these two tyrosines in the second half of the tyrosine kinase, and also these two tyrosines in the C-terminal tail. Okay, and the tyrosine kinase of receptor 2 will reciprocate. It will phosphorylate the tyrosines on receptor 1, basically. Okay, so you're going to create loads of phosphorylated tyrosine residues, and these are known as phosphotyrosine residues. Okay, and it's these phosphotyrosine residues that are on the cytoplasmic domain of these vascular endothelial growth factor receptor 2s that are now going to take this pathway further. They are what are going to trigger the downstream pathway. Okay, so we will begin with the PI3 kinase AKT-mTOR pathway, but in fact, there are absolutely loads of pathways downstream of the vascular endothelial growth factor receptor 2. So these phosphorylated tyrosine residues, these phosphotyrosine residues, are going to activate a whole myriad of downstream pathways, but we will start with the PI3 kinase pathway. Okay, so what's going to happen is an enzyme that has a very special domain which is capable of binding to phosphotyrosine residues is going to come up and bind to the phosphotyrosine residues on the cytoplasmic side of these vascular endothelial growth factor receptor 2s. Okay, so I'll draw this here. So this enzyme is known as phosphatidylinositol free kinase. Okay, and for short, phosphatidylinositol free kinase is often abbreviated to PI for phosphatidylinositol and then free kinase, or even shorter, it can just be abbreviated as PI3K. Okay, so P for phosphatidyl, I for inositol, free for free, and then K for kinase. That's how you would write it, but you wouldn't read this as PI3K. If you were to read this in short, you'd say PI3 kinase, so you'd abbreviate phosphatidylinositol to just PI3. Okay, so PI3 kinase, basically it has a very special domain which is capable of binding to phosphotyrosine residues. So I'll highlight this special domain in turquoise here. And this special domain is known as an SH2 domain. Okay, and SH2 stands for Sark homology 2 domain. Okay, so this is a Sark homology 2. So the S is for Sark, H is for homology, and 2 is for 2. Okay, so it's the SH2 domain, the Sark homology 2 domain. So it's a domain that is very similar to a domain that you have within the protein Sark, which stands for steroid receptor coactivator. Okay, now, this conserved domain is special because it can bind to phosphotyrosine residues. So once you have these phosphotyrosine residues on the cytoplasmic side of your vascular endothelial growth factor receptor 2s, what will happen is the Sark homology 2 domain of the phosphatidylinositol free kinase will bind to them, and that is going to sequester this enzyme at the inner leaflet of the phospholipid bilayer. So let me show it here. It's now come and docked on here with its special domain here. And what does that do? It means that the enzyme is just underneath the cell membrane, basically, and the target for this enzyme is within the cell membrane. So this is why binding it there is going to lead to it having its effect, because now it's in contact with its substrate. Okay, so what is the substrate for phosphatidylinositol free kinase? Well, it's a molecule known as phosphatidylinositol 4,5-bisphosphate. So, for short, phosphatidylinositol 4,5-bisphosphate is abbreviated to PIP2. Okay, so again you abbreviate the phosphatidylinositol to PI. okay? And then the next abbreviation is that you're going to abbreviate phosphate to P, and then because you've got two phosphate groups, you'll just put a 2 down there. So PIP2 is short for phosphatidylanocetyl 4,5-bisphosphate. Okay, and often people will just pronounce that as PIP2. Right, so phosphatidylanocetyl 4,5-bisphosphate, basically it's a normal component of the phospholipid bilayer. In all of the cells of your body you will have phosphatidylanocetyl 4,5-bisphosphate molecules in your cell membranes basically. So, I want to show you a cartoon of its structure to convince you that really it's just a modified phospholipid. Okay, so we'll start off with the structure of a normal old, boring old phospholipid. Okay, which is sometimes known as a phosphatidate molecule. Okay, so phosphatidate is the strict name for a normal old, boring old phospholipid. Right, so let's go over this structure. So, these vertical lines, which I'm now colouring in orange, these are going to represent the long-chain carboxylic acids. And by the way, I've drawn these two lines to represent the lipid bilayer of the cell membrane. Okay, so these vertical lines, which I've coloured in orange, these are going to represent the long-chain carboxylic acids. And we have two of these that are esterified to the first and second alcohol groups of the glycerol molecule. Okay, and another name for long-chain carboxylic acids is they're also sometimes called fatty acids. Okay, so the biochemist's name for long-chain carboxylic acids is fatty acids. The more correct name is long-chain carboxylic acids. So these are carboxylic acid molecules which have an extremely long tail, basically, and which is generally very hydrophobic and interacts, therefore, well with other hydrophobic molecules. Okay, and will interact, therefore, very nicely with the hydrophobic core of the lipid bilayer here. Now, this other molecule, this horizontal line that I've now coloured in green, this represents the backbone of the phosphatidate molecule. This represents glycerol. And again, glycerol is the old biochemist's name. The more correct name for this is propane 1,2,3-triol. And although propane 1,2,3-triol is more of a mouthful than glycerol, it's a useful name because it tells you exactly what this molecule is. It's a 3-carbon hydrocarbon where you have alcohol groups coming off the first, the second, and the third carbon. Okay, so every single one of the carbons has an alcohol group coming off it. So off the first and second carbon's alcohol groups, you have these long-chain carboxylic acids esterified. And off the third carbon's alcohol group, you then have a phosphate group, and this is again attached via a phosphoester link. Okay, so the phosphate group has bound to the alcohol group via a phosphoester link. Okay, so when people talk about phospholipids, this is probably what you think of, a phosphatidate molecule. This is a normal old, boring old phospholipid. But there are other modified versions of this, okay? And phosphatidylanocetyl 4,5-bisphosphate is just a modified version of phosphatidate, basically. That's where this phosphatidyl comes from. It basically means a phosphatidate molecule, where you're viewing this now as a group, basically, that's attached to another molecule. Okay, so basically what you're going to do is attach on an inositol molecule onto this phosphatidate molecule, and the way you're going to do it is that this phosphate group here that's attached onto the third carbon's alcohol group of the glycerol molecule, this has another free alcohol group. Look at this phosphate group here. Even once we have used one of the alcohol groups up in a phosphoester link, you've still got this other alcohol group, and you've still got the phosphorus atom doubly bound to an oxygen atom there. So this is still analogous basically to a carboxylic acid group. And indeed you can use that to make another phosphoester link. And you're going to link it to another alcohol group which is on the inositol molecule. So inositol is a 6-carbon ring basically, a 6-membered carbon ring. Okay, so it's basically the proper name for inositol is cyclohexane 1,2,3,4,5,6 hexol. Okay, and what that means is that basically you've got this six-membered carbon ring where all of the bonds between the carbon atoms in this ring are single bonds. And therefore each of the carbons in this ring has two spare bonds that you need to attach something to. And each carbon is given an alcohol group and each carbon is given a hydrogen. So that means that you have 6 alcohol groups in total coming off this inositol ring, one off all of these 6 carbons basically. And as I say, the proper name for inositol is cyclohexane, which is the name for this 6-membered carbon ring, and then 1, 2, 3, 4, 5, 6, and then hexol. Okay, because you've got an alcohol group coming off every single one of the carbon atoms in the cyclohexane ring. Right, now what you're going to do is you're also going to, well actually let's just discuss what this molecule that we've created now is. This molecule is what you would call phosphatidyl inositol. This is PI. We've got a phosphatidate molecule linked to an inositol molecule, but we want phosphatidyl inositol 4,5-bisphosphate. So what we need to do is link 2 more phosphate groups off the 4th and the 5th carbon of the inositol ring. So basically, there are many different isomers of inositol. There are actually 9 different optical isomers of inositol, because if you think about it, if you imagine having all of these 6 carbons in the plane, then when you're sticking the hydrogens and the alcohol groups off every single one of the carbons, either the alcohol group can go into the page and the hydrogen can come out of the page towards us, or it can be the other way around. The alcohol group comes out of the page towards us and the hydrogen goes into the board, into the page away from us. So there are many different ways that you can orient the alcohol groups. For instance, they could all be on the same side. They could all be going into the page away from us, and all the hydrogens could be coming out of the page towards us, or it could be half and half. So there are many different optical isomers. The optical isomer that is usually used in cells is called myo-inositol. Okay, so very strictly, if you ever hear people talking about biological molecules which contain inositol, strictly you'll see the 3-letter word myo in front of the inositol, which will tell you which optical isomer you're dealing with, but often people just will drop that. Okay, but strictly, if you ever do see that, that's what it is referring to. It's referring to the specific optical isomer of inositol that is usually found in biological systems. Okay, now, basically, myo-inositol has a certain way of labelling up the carbon atoms of the ring, okay, and this one up here that is attached to this phosphate group turns out to be the first one, and then this one will be the second one here, this one the third one, here's the 4th one, so we'll stick a phosphate group off this one, so that we've got the 4th, and then off the 5th one, you'll also have a phosphate group like so. Okay, so this creates phosphatidyl inositol 4,5-bisphosphate basically. Okay, and as I say, you might look at that and think, why isn't it called phosphatidyl inositol 3,4-bisphosphate? Why haven't we called this carbon the 3rd carbon? And as I say, it's to do with the way that the carbons of this specific optical isomer of inositol are labelled. You have the phosphate group coming off the 5th carbon of myo-inositol, not the 3rd carbon. Okay, right. So this is the structure then of phosphatidyl inositol 4,5-bisphosphate, and hopefully what I've convinced you of is that basically it's just what you would imagine as a phospholipid with a bigger head. It's just a modified phospholipid, a modified phosphatidate molecule. Okay, so this is a normal component of the phospholipid bilayer, and it's just got, a bigger head which sticks into the cytoplasm. And this is the target for the enzyme phosphatidylainositol-3 kinase. So once the phosphatidylainositol-3 kinase has bound to the phosphotyrosine residues on the cytoplasmic side of the vascular endothelial growth factor receptor 2, what will happen is it will set to work on these phosphatidylainositol 4,5-bisphosphate molecules within the phospholipid bilayer, and it's going to add phosphate groups onto the third carbon of the inositol ring. So what it's going to do is it's going to add an extra phosphate group on here, like so. Okay, now what will that create? Well, the molecule that this creates is what is known as phosphatidylainositol, because you've still got the phosphatidate molecule, you've still got the inositol, okay, but now you have 3 phosphate groups coming off the inositol ring of the phosphatidylainositol molecule. Okay, so this will now be phosphatidylainositol 3, because the first phosphate group comes off the third, then 4, then 5, and now tris phosphate, because we've got 3 phosphate groups. Okay, and for short, phosphatidylainositol 3,4,5 trisphosphate is often abbreviated to PI-P3. So again, the first P is for phosphatidyl, the I is for inositol, the second P is for phosphate, and because we've got 3 phosphate groups, you put a 3 there. So basically, this PI3 kinase is going to convert PIP2 into PIP3. Okay, and now this PIP3 is going to be responsible for the next stages in this pathway, and we'll continue this discussion in the next video.